In this video, we will discuss the reservoirs of infectious disease, differentiate contamination and infection, and explore the portals of entry and adhesion factors utilized by pathogens. The majority of pathogens cannot exist outside their host for a prolonged period of time. In order for the disease to continue to exist, the pathogen must have a place where it can be maintained as a source of infection. The site where the pathogen is maintained as a source of infection is known as the reservoir. There are three types of reservoirs of infectious disease of humans. They are animal reservoirs, human carriers, and non-living reservoirs. Animals can act as reservoirs for diseases that can be spread to humans. Diseases that can spread from an animal host to humans are known as zoonoses. Zoonotic diseases can be acquired from animals in a variety of ways. Some examples include the direct contact with an infected animal, consumption of food or fluid contaminated with urine or fecal material, the bite of an insect vector, or direct contact with animal waste. Those more likely to contract a zoonotic disease are those who are more frequently exposed to animals like veterinarians, forest rangers, zookeepers, and pet owners. Over 150 zoonoses have been identified. The table shows some examples of zoonotic diseases, their causative agent, the animal reservoir, and mode of transmission from reservoir to human host. The human is typically a dead-end host, meaning that humans do not serve as a significant reservoir for the reinfection of animals. This is because of the manner in which the pathogen is transmitted from one host to another. For example, those organisms that must be consumed through contaminated tissues, such as muscle tissue, are not likely to pass from human to animal because animals do not typically consume humans, and do not move from one human host to another because we do not practice cannibalism. The most common way a pathogen would move from a human to an animal is by transmission of blood-borne pathogens. Humans can also act as reservoirs for pathogens. Some individuals will develop an illness, and while they are carrying the organism, they can serve as a source of infection to spread the pathogen to other human hosts. Other individuals may carry a pathogen and never develop any signs or symptoms of an illness. Such individuals are known as carriers. Humans can act as reservoirs for the causative agents of AIDS, typhoid fever, hepatitis, and gonorrhea, among others. Soil, water, and food can serve as non-living sources of pathogens. Typically, these non-living reservoirs serve as sources for infection due to contamination with fecal material or urine. Food that has not been properly prepared or stored can also act as a reservoir for pathogens. Before we begin a discussion regarding infection, we first need to accurately define infection and differentiate infection from contamination and disease. Contamination refers to the mere presence of microorganisms in or on the body. Since microorganisms normally reside in many areas of the body, the simple presence of a microorganism does not mean that one has an infection. Infection occurs when microorganisms enter the body and begin to multiply. Infection is differentiated from disease in that the disease occurs when any part of the body's normal function is disrupted. Infection and disease are frequently used interchangeably by many, but they are not the same thing. The HIV virus is the cause of the disease known as acquired immunodeficiency syndrome. An individual can be infected with the virus and show no sign or symptom of disease. HIV viral numbers increase in the host and begin to cause disruption of the body's ability to defend itself. At that point, the infection has caused disease. It is important to note that infections may or may not result in disease. In order for an organism to cause infection, it must enter through the correct site on the body and must successfully attach to the host. For the remainder of this lecture video, we will discuss the sites by which pathogens enter the body and the mechanisms utilized to attach to their host. The route through which a pathogen enters the body is known as a portal of entry. Portals of entry include the skin, mucous membranes, placenta, and parenteral route. The skin is composed of many layers of cells that typically act as a barrier to prevent the entrance of most pathogens. However, if the integrity of the skin is broken, infection can easily be established. The integrity of the skin can be broken in a number of ways, including, but not limited to, cuts, scrapes, abrasions, surgical sites, and burns. Some pathogens do not require a breach in the skin's integrity. They may be capable of entering through hair follicles or sweat gland ducts, or by directly penetrating the skin. Many pathogens gain access to the body through the mucous membranes. The mucous membranes are found lining the respiratory tract, gastrointestinal tract, genitourinary tract, and conjunctiva of the eye. These regions provide a moist, warm environment that is hospitable to many microorganisms, including pathogens. The respiratory tract is the most common portal of entry for pathogens. Pathogens can be inhaled through the nose or mouth on droplets of moisture or on dust particles. There are many viruses that can enter into the respiratory tract just by making contact with the conjunctiva of the eyes, and then are washed into the nasal cavity by tears. The causative agents of influenza, the common cold, measles, strep throat, smallpox, tuberculosis, and pneumonia are a few examples that utilize the respiratory tract as their portal of entry. The gastrointestinal tract also acts as a portal of entry for many pathogens. Pathogens that utilize the GI tract to enter the body must be equipped to withstand the hydrochloric acid of the stomach and the many enzymes of the intestines. Those capable of surviving can establish infection and disease. A number of parasitic protozoans, helminths, bacteria, and viruses enter through the GI tract. This portal is also the route by which prions may gain access to the body. The causative agents of diseases such as hepatitis, typhoid fever, amoebic dysentery, and cholera enter through the gastrointestinal tract. The genitourinary tract is also a portal through which organisms enter the body. Some of the pathogens require a break in the mucous membrane, while others are capable of penetrating unbroken mucous membranes. The causative agents of sexually transmitted infections can enter through unbroken membranes. Sexually transmitted pathogens are associated with diseases such as AIDS, genital warts, gonorrhea, syphilis, and herpes. The placental membrane is generally a protective barrier for the developing embryo or fetus. However, there are some pathogens that are able to breach the barrier to cause infection of the embryo or fetus. This may lead to devastating consequences such as spontaneous abortion, birth defects, or premature birth. Among the pathogens capable of crossing the placental membrane include the causative agents of syphilis, AIDS, the German measles, and toxoplasmosis. The route known as the parenteral route is not a true portal of entry, but rather a way to bypass the other portals. Pathogens that enter via the parenteral route are deposited directly into the tissues beneath the skin. Pathogens may enter via the parenteral route in a variety of ways, including punctures, insect bites, stab wounds, surgical procedures, deep abrasions, and injections. The causative agents of tetanus, gangrene, malaria, African sleeping sickness, and Chagas disease are some of the pathogens that employ the parenteral route. Microorganisms wishing to establish themselves on or in the body must have some means of attaching to their host. Attachment is also referred to as adhesion. Structural features or special attachment proteins that permit a microorganism to adhere to a host are called adhesion factors. Adhesion factors are considered a virulence factor because pathogens must first adhere to their host before they can begin to multiply and infect the host. Microorganisms lacking adhesion factors cannot establish infection. Thus, the first step in infection is adhesion. A variety of adhesion factors in the form of specialized structures and adhesion proteins exist on the organisms that invade the human body. Bacteria and viruses have lipoproteins and glycoproteins, also known as ligands, on their surfaces that allow them to attach to receptors on host cells. The types of cells to which they can adhere is limited by the interaction between the ligand on the surface of the virus or bacterial cell with a receptor on the host cell. If a receptor is not present, they cannot adhere. Therefore, viruses and bacteria are restricted to particular cells or regions of the body. Changing or blocking the binding site for the ligand attachment can prevent infection, thereby also preventing disease. Other pathogens may utilize specialized structures to attach to their host. Parasitic protozoan helminths have structures on the surface of their cells that permit attachment. Protozoans may use adhesion discs, while helminths may employ suckers or hooks for adherence. Biofilms are another means of attachment for microorganisms. The microorganisms within the biofilm secrete sticky substances that allow the community to adhere to a surface.